Welcome to Module 6 of Sensation and Perception Online. This presentation will cover the types of somatosensation, the somatosensory receptors, the two major somatosensory pathways to the brain, and how somatosensation is organized in the brain. The somatosensory system includes the detection of changes in a variety of energies. The primary sources are pressure, which corresponds to tactile perception, kinesthesis, which involves sensing where our limbs are and what they are doing, temperature, and pain. There are two major nerve pathways which transmit sensory information to the brain. The first is the dorsal column medial lemniscal, or DCML, pathway, which transmits tactile and kinesthetic information. The other is the spinothalamic pathway, which transmits temperature and pain information to the brain. We will now look at each of these topics in more detail. Tactile receptors, like the hair cells of the auditory system, are mechanoreceptors. What this means is they respond when they are physically moved. Tactile receptors respond to being squished and stretched. That is, they respond to pressure on the skin and distortions of the skin. They are found in the upper layers of the skin. There are four types of tactile receptors. The Meissner corpuscles are located near the skin surface and detect changes in skin deformation and low-frequency vibration. The Merkel cell complexes are also near the surface of the skin and detect sustained pressure and are involved in texture perception. The Pacinian corpuscle is the deepest in the skin and detects skin deformation and high-frequency vibration. The Pacinian corpuscle looks a bit like an onion, having many layers. Unlike an onion, these layers are easily distorted when pressure is applied to the skin. When the pressure is removed, it returns to its original shape. The Ruffini endings are also deeper in the skin. They detect sustained pressure and skin stretch. They are involved in detecting finger position and grasp stability. I'm sure we've all wondered how sensitive the skin is to touch, that is, how much pressure is necessary for a touch to be detected. As kids, we may have even tried to touch someone without them noticing. We may even have some idea, in fact we probably do have some idea, as to which parts of the body are the most sensitive. That is exactly what this figure is going to show us. Before revealing the figure, which parts of the body do you think are most sensitive to touch? Which parts of the body will be least sensitive? Let's see if you're right. Did you predict that the upper lip and nose would be the most sensitive parts? How about that the sole and big toe, the hallux, would be the least sensitive? Or even that the fingers would be less sensitive than the back or belly? If those numbers seem strange, those sensitivity numbers, it might be due to confusion between sensitivity and acuity. Sensitivity is just the ability to detect pressure. Acuity is the ability to detect detail, like textural patterns. The way to assess acuity is to find the 2-point threshold. That is the minimum distance between 2 points at which they are still perceived as 2 points and not one. This is tested by simultaneously touching 2 points on the skin at the same time and with the same pressure. Perhaps you have been in a boring meeting with a sibling or a friend, and you diverted yourselves by taking turns drawing letters on each other's back with your finger, having to guess which letter was drawn. The smaller the letter is drawn, the more difficult it is to guess. Let's look at the areas that are most sensitive to detail, as measured by the 2-point threshold. Now we see that the fingers and thumb are the most sensitive, followed closely by the upper lip, cheek, nose, and big toe. The calf, shoulder, back, and breast have the highest 2-point thresholds, meaning two simultaneous touches have to be farther apart, about 40 millimeters or 1.5 inches, to be detected as two touches and not one. The index finger, on the other hand, can detect 2 points as two when they are as close as 1/8 of an inch apart. Kinesthetic sensation is the detection of limb movement and location. This is a sense that is often ignored or taken for granted, but it is critical for our ability to do the simplest task, like sitting up. Think of how many movements we make without watching what we are doing. The way we are able to do them is because we can sense what we are doing as we are doing it. The receptors for kinesthetic sensation are muscle spindle sensory fibers. These are found in the intrafusal muscle fibers, the inner muscle fibers. The type 1A sensory fiber detects changes in the length of the intrafusal muscle. It is fast adapting, meaning that it stops firing when the muscle stops moving. These fibers provide information about the speed and force of muscle movement. Type 2 sensory fibers continuously report the length of the intrafusal muscle. They are slow adapting, meaning that they fire continually depending on the length of the intrafusal muscle. These fibers let us know exactly where our limbs are at any given time. There are also sensory fibers in the tendons and joints. These provide information about the tension of muscles on the tendons and also about the extension of a joint. In both cases, these fibers provide a protective function. Thermoreceptors are sensory receptors that respond to changes in skin temperature. There are two types of fibers, cold fibers and warmth fibers. Cold fibers fire more as the skin temperature drops to about 77 degrees Fahrenheit, which is where the firing rate peaks. Warmth fibers, on the other hand, fire more as the skin temperature increases, peaking at about 113 degrees Fahrenheit. As you can see, the point where the two curves cross corresponds to normal human body temperature. That means that these fibers constantly monitor the skin temperature and provide feedback whether the skin temperature gets too cold or too hot. That is, they provide feedback to keep skin temperature in the Goldilocks zone, just right. Lastly, thermoreceptors are change detectors. A smooth metal object always feels different than a smooth wooden object. Why? Because metal objects conduct heat from our skin. That is, they cool our skin much more than wooden objects do. The temperature changes of the skin that occur when we touch objects are an important part of tactile perception. Nociceptors are pain receptors. Think of it as noxious receptors. They are receptors that respond to noxious stimuli. These are primarily referred to as bare nerve endings, as they do not have a corpuscle or a complex body surrounding them, like the tactile receptors do. These receptors respond to extreme pressure and other types of tissue damage. There are two main groups of nociceptors. The first are A delta fibers. Of the two types, these are the faster transmission fibers, and they respond to intense pressure, including cuts and tears, and to cold. The others are C fibers. These are slow transmission fibers, and they respond to warmth, itch, muscle burn, and cramp pain. It may be surprising that the warmth fibers, which respond to burns, are slow fibers. Don't we respond very quickly to a hot surface? Yes, we do, but that is because of a built-in spinal reflex. A hot stimulus on the index finger is detected by a receptor with its cell body in the dorsal root ganglion near the spinal cord. This sensory neuron synapses with a small interneuron in the spinal cord, which synapses with a motor neuron, which causes the arm muscle to contract, pulling the finger away from the heat. All of that happens before the brain even detects that a hot stimulus was sensed. These two types of fibers, each with a different speed, helps us to understand the two-phase process of pain. The first wave of pain is the sharp, fast pain. As it subsides, we then feel the throbbing, slow pain. A delta fibers, fast, sharp pain. C fibers, slow, throbbing, warmth pain. Now let's look at the pathways to the brain. The first is the dorsal column medial lemniscal pathway, or DCML for short. The DCML consists of 3 neurons. The receptor axon runs from the site of activation all the way to the brain stem. That is, from finger touching something and feeling it, that neuron that detects that runs from the tip of your finger all the way to the medulla in the brainstem. There are synapses on another neuron. That brainstem neuron, its axon extends to the opposite side of the body and then to the thalamus. Finally, the thalamic neuron runs to the primary somatosensory cortex in the parietal lobe. Some characteristics of this pathway: this is carrying tactile and kinesthetic sensation. It transmits rapidly. This is a fast transmission pathway. Now, partly it's fast transmission because one neuron runs from the site of activation all the way to the brainstem. But that's only part of the story. The other part is that these are wide fibers and they're myelinated fibers. So they're fast. In the brain, so in the primary somatosensory cortex, the sensations are somatopically mapped. That is, adjacent areas of the body are connected to adjacent areas of the brain. We'll talk more about this in a couple of slides. The second pathway is the spinothalamic pathway. This pathway also involves 3 neurons. The receptor neuron, its axon runs from the site of activation to the spinal cord. In the spinal cord, it synapses with another neuron, and there in the spinal cord crosses to the opposite side of the body, and that axon runs to the thalamus, and from the thalamus to the somatosensory cortex. So some similarity between these two pathways, both run to the thalamus. The difference is where the crossing over takes place. For the spinothalamic pathway, the crossing over takes place at the spine. For the other, it takes place in the medial lemniscus, or the brainstem. Characteristics of the spinothalamic pathway. First, it's carrying pain and temperature sensations. Second, this is the slow transmission pathway. Right? Pain and temperature, these, even though the A delta fibers were referred to as being faster fibers, they're only faster than the C fibers. They're still not as fast as the DCML fibers. The mapping also to the primary cortex is less precise, which is why it's a little harder to tell exactly which part of the body hurts. Right? You might feel that something itches, but you can scratch around in different places, and that can help to soothe the itch. You don't have to scratch right on the point. Sometimes we can feel a pain somewhere, and it might actually be hard to describe where that is. But when you apply pressure on the right point, haha, you found it. It hurts right there. There's less precise mapping of pain and temperature sensation. The somatosensory cortex is found in the parietal lobe along the central sulcus, which divides the parietal and frontal lobes. The motor cortex is on the opposite side of the central sulcus in the frontal lobe. Remember that when we discussed the DCML and spinothalamic pathways, we mentioned that the fibers crossed over to the opposite side of the body. That means that somatosensation is contralaterally represented. That is, the left side of the body is sensed by the right hemisphere of the brain, and the right side of the body is sensed by the left hemisphere of the brain. Other characteristics of the somatosensory cortex include somatopic representation and cortical magnification. These should sound somewhat familiar. Somatopic representation is akin to topographic representation discussed when we covered the primary visual cortex. What it means is that adjacent areas on the skin have connections to adjacent areas in the brain. It can be said that the brain contains a sensory map of the body, often referred to as a homunculus or little man. Notice, though, that the somatopic map is distorted. The fingers have much more cerebral area dedicated to them than the legs and other large body parts. As you recall, this is referred to as cortical magnification. The tactile receptors that respond to fine details, high acuity, have more brain area dedicated to them. Recall back to the slide that showed the 2-point thresholds. The areas with the smallest 2-point thresholds are the areas with the largest amount of dedicated cortex. To review, the somatosensory system involves many sensations: pressure, kinesthesis, vibration, temperature, pain. The somatosensory receptors are mechanoreceptors. There are mechanoreceptors in the skin that give us sensitivity to touch, to pressure, and acuity, detecting detail. And that varies across the body. That is, the sensitivity and acuity varies. There are mechanoreceptors in the muscles, joints, and tendons that let us know how our arms, how our limbs are moving, and also where they are at any given point. The receptors in the joints and tendons let us know about how much force muscles are putting on the tendon and the position of the joints so as to not to hyperextend them. Thermoreceptors come in two classes: warmth fibers and cold fibers. And nociceptors detect pain, itch, etc. There are two major somatosensory pathways: the fast, the DCML pathway for mechanoreceptors, and the slow spinothalamic pathway for temperature and pain. The somatosensory cortex has two really important characteristics. It is somatopically mapped, that is, we have a little sensory homunculus, the body is mapped onto the brain, and there is cortical magnification, areas of high acuity, that is, body parts with high acuity have more cortex devoted to them.